Hi everyone, this is Jonathan Cedillo, student at Grand Canyon University. Today I'm presenting to you an analysis of action potential parameters. Presentation for class Biology 515, Concepts of Human Physiology 1, Professor Sandra Barrera. Parameters of action potentials. Cortical neurons have action potentials different from each other. In the following paper, which is the focus of this presentation, the action potential of neurons will be analyzed. The analysis of neural parameters encompasses the width, height, and energy of actual potentials together with their spiking frequency and inner spike intervals, or ISI, which were studied from 91 human samples of cortical neurons. Authors of this article claim that smaller actual potentials with low energy have more abundance of spiking frequency in comparison to larger action potentials. In addition, the article states that different actual potentials, including their spike in frequency and specifications, can promote similar excitatory responses. This information is analyzed using computational data. The authors of the paper claim that the change of actual potentials activity can be of importance when used during cognitive applications. Tools for cognitive applications. Spiking neural networks can be an important tool used for the engineering of cognitive applications. However, there has been a lot of research in the understanding of how neurons work in the brain to implement these applications. Other studies compare the properties of actual potentials and claim that these differences are key physiological features. In the paper of discussion, Actual potential parameters and spiking behavior of cortical neurons, a statistical analysis for designing spiking neural networks, work provided by Chakraborty et al. A detailed criterion is given to correlate the spiking kinetics of human cortical neurons to the respective electrophysiological descriptions. The focus was the activity of actual potential characteristics in the activation of spiking neural networks to understand a general view of how the network of interest behaves. For this, 91 neurons were tested. Such neurons provided between 5 to 15 spikes that range from 70 picoamperes to 200 picoamperes. The following properties, together with the correlation with height and energy per spike activities, were also analyzed. Experimentation phase. The paper attributes that important data was identified and extracted from the database AIBS. Continuing, with the aid of a probe, electricity was run over neurons to measure the activity of membrane potentials with high precision electrodes. Membrane potential control was set to a 50 kilohertz and filtered by a 10 kilohertz Bessel filter. In figure 1A, which is on the left side of the screen right here, the stimulus of a neuron with a 90 picoampere pulse for 1 second is presented. The membrane potential is estimated to be negative 62 millivolts in the absence of an electrical stimulus. After the application of the 90 picoamperes, membrane potential rises, which resulted in 8 spikes, as you can see in figure 1A. 1, 2, 3, 4, 5, 6, 7, 8, right there. Figure 1B, which is just down below right here, is a representation of a common actual potential. TAP, right here, is the time an actual potential takes to occur. Following, depolarization is the next step. After the membrane potential reaches climax, it repolarizes, bringing the cell to a normal state. The time an actual potential takes to occur has been split into the time it takes for the climax and the time it takes to go back to normal or falling time, which is labeled as TF, which is right here. By adding these two components, the result is the time for sodium ion channels to open and depolarize the neuron. During the time that it takes the cell to go back to normal, membrane potential, voltage-gated potassium channels open to repolarize the neuron. Description of spiking in 1 second, or its frequency, an inner spike interval with the said stimuli were also recorded. In the same experiment, the 91 chosen neurons were statistically analyzed. In Table 1, neuron cells were accommodated the following way. Neurons with 5 to 15 spikes at 70 to 90 picoamperes were labeled as S1. Neurons with 5 to 15 spikes at 110 and 130 picoamperes were labeled as S2. Neurons with 5 to 15 spikes at 170 to 200 picoamperes were labeled as S3. The cells were stimulated for 1 consecutive second. The two currents applied for that 1 second that, that were the low and high excitations, which have a difference of 100 picoamperes, as shown in Table 2. Data shows that cells in all groups have similar kinetics during the low excitation stimuli. Also, the spiking kinetics of cells in all groups were different at the high excitation stimuli in contrast to the low excitation stimuli. Table 2 is a representation of the correlation between actual potential duration and spiking frequency. Capital N refers to the spiking frequency of neurons, which is right here. And let me just point it out with the pen, right here. And the column right next to it is the number of actual potentials fired in 1 second. Scientists reveal that the time for an actual potential to occur plays a critical role in the spiking frequency. Within the table, clusters of neurons that behave similarly have been grouped together. In Table 2, the neurons stimulated at low excitation from all groups presented a similar number of spikes. However, when increasing the stimuli, cells behave differently. Figure 2 represents the time an actual potential takes to occur versus the number of spikes created in 1 second for all groups S1, S2, and S3 with their respective cluster groups and electric current stimuli. Notice that the more time it takes for an actual potential to be initiated, the fewer spike-fired neurons will be produced in all groups. Probability of density function of inner spike intervals. From the data collected, it has been concluded that the neural activity of clusters is different in their spiking frequency. In consequence, their fall time, which is when membrane goes back to normal, when the membrane potential goes back to normal, also different from each other. In addition, it has been revealed that the fall time of an actual potential is lower for those neurons that promote more spikes in comparison to those neurons that promote fewer spikes. This is suggesting that the actual potential time and falling time affect the spiking frequency of a neuron. With previous data, the authors of the paper stated that neurons belonging to any of the cluster groups have a like spiking activity when similar stimulus is applied. Interestingly, in Figure 3, which is the figure that I'm showing right now, the probability of the density function of the inner spike intervals was analyzed, and it showed results of deviation depending on the low or high excitation or stimuli applied to the neuron clusters. As we can see, a low excitatory stimulus in all clusters is similar. C1 and C2 at a high excitation, in Figure 1, are close to each other. However, that is not the case for cluster K1 and K2. Also, it's not the case for clusters Q1 and Q2. As the curves in the graph show, a higher peak, the excitatory stimulus is also high, making the probability density function slopes even steeper. Before it has been discussed that neurons at low excitatory stimulus produce between 5 to 15 spikes in clusters C1 and C2. On the other hand, the spiking frequency is completely different in the presence of a greater stimulus. This can be seen in the inner spike intervals presented in graphs A, B, C of Figure 3. Height and energy of action potentials. The kernel density technique was used to analyze the height of action potentials. Figure 4, we can see the computational results of this technique. For example, the height of the actual potentials of cluster Q2 is greater than Q1, with a narrower but with more spiking frequency. Similarly, the actual potentials in graph A and B present a similar pattern. It has been established that the time it takes an actual potential to reach its maximum height is larger for neurons with wider actual potentials. Thus, wider actual potentials are higher than narrow actual potentials. In addition, the previously mentioned neurons demonstrated a low firing rate in comparison to neurons firing smaller actual potentials. Continuing, it is predicted that neurons with wide and tall actual potentials would deplete more energy for every spiking activity, and the opposite is true for narrower and shorter action potentials. To conclude, in this study, Actual Potential Parameters and the Spiking Behavior of Cortical Neurons, a statistical analysis for designing spiking neural networks by Chakraborty et al., the authors carry out a statistical analysis of the actual potential activities of 91 neurons. With this same analysis, It has been determined that neurons with narrow and short action potentials show more spiking activity than neurons with wider and taller action potentials when a strong excitatory stimulus is applied. In addition, neurons showing more spiking activity deplete low energy for every action potential they produce. Also, the change in excitation demonstrated that the information gathered is larger for neurons with narrower and shorter action potentials with abundant frequency. This was retrieved from the stimulation of neurons and processing of that activity using computational technology. Finally, it is suggested that the knowledge and information acquired after this study will be of importance for the engineering of spiking neural networks in cognitive applications. Thank you for watching. See you next time.